Hello everyone. My slides are visible and am I audible?
Yes, ma'am, you are audible.
Okay. I was facing actually some technical glitch at that time. I was not able to join. So let's start. So we have to start today the module 2, that is microbiome and human health, and we will just go into more detail because this is an intermediate level. We have gone through all the basics in the module 1, so we will start little bit of more deeper and we will see what is going on. So firstly we will see this part, role of gut microbiota in digestion and nutrient absorption. This is important. We have to understand that how the gut microbiota microorganisms play a vital role in the digestion and the different kind of nutrient absorption. So after that, we will see these three parts like gut brain axis, role in immunity, and dysbiosis. So first of all, we all know that vitamins and minerals, which are collectively termed as micronutrients, which are required in less quantity, are very crucial for human health. It is very obvious thing, but these micronutrients are degraded or not synthesized in an adequate amount if these microorganism is somehow altered. So these micronutrients are essential. What is the role of micronutrients in our body? Many of the micronutrients act as a coenzyme for different functioning of different vital enzyme in our body. So they are the essential co-regulator of fundamental biosynthetic cellular reaction such as immune and energy function, as well as growth, bone health, fluid balance, and other biological process. So in short, there are so many different kind of role of each and every micronutrient. Some are playing role as a coenzyme, some are playing role as a catalyst to complete the reaction speedily, and also in providing immunity and in growth and normal maintenance and repair of our body cell. So we will see micronutrient deficiency in India and all over the globe is very common. 4 out of 5 people are facing micronutrient deficiency, but they don't consider it as a uh, very dangerous thing. But slowly it may be dangerous, it may be fatal, because prolonged micronutrient deficiency will result in so many different kind of cellular irregularities, cellular deficiencies, and ultimately it may lead to severe metabolic diseases. So micronutrient deficiency is considered a global health issue as it is associated with severe health problems, particularly in children, because the diet of children, we can see they only eat uh, like they don't eat any vitamin rich or any mineral rich food, like they don't like fruits, they don't like green vegetables, which is very much uh, beneficial for having vitamins and minerals. So we have to take care of the diet which is rich in micronutrients. So if we are not having micronutrient in an adequate amount in our body, it may leads to mental retardation. We can see so many different kind of mental abnormalities. So the deficiency of micronutrient like vitamin D may contribute to disordered immune response both in children and adult. Vitamin D deficiency is also very common in adults too. So this picture shows the micronutrient inter-exchange between the gut microbiota and the host. Suppose if a normal host is there, this section, if you can, this circle shows the interaction with whom? Interaction with the host and the microbiome. So you can observe this left half, this portion is of host, and this right portion is of microbiome. So how to know that what the microbiome is doing in our body, what role are they playing, what crucial function they are playing there. So you can see there are so many different groups available in this microbiome, and they are responsible for the synthesis of different kind of vitamin, minerals, and also different kind of micronutrients. So you can see, if your gut microbiome is normal, they don't have any kind of infection, they are not altered, so they will synthesize, like they will synthesize each and every micronutrient, vitamins, and short chain fatty acid like butyrate, propionate, acetate. Everything will be synthesized in an adequate amount. Whatever amount is required for the normal functioning of the body, they will synthesize, because their presence is not altered, their environment is graded, they are normal, so they will perform their metabolic machinery, and they will synthesize for what they are designated as. If they are designated to synthesize some vitamins, if you are providing normal condition, they will synthesize. And secreted microbiome metabolite contribute to host micronutrient status. So if you are having normal microbiome, if you are having normal microbiome, all the microorganisms are in a very good state, then they will show their metabolism, their metabolic machinery will be very good, and after that they will synthesize some metabolite like short chain fatty acid, fat soluble vitamins ADEK, water soluble vitamin B1 to B12, and also vitamin C. As you all know, ADEK are fat soluble and B and C are water soluble vitamins. So all these things will be synthesized only when the microbiome is not disturbed. So you can say, if a person is not having good gut, he or she will suffer from micronutrient deficiency. Micro Micronutrient deficiency. I don't know what is happening. If a person is not having good gut, it means somehow he or she have disturbed their own microbiome composition. Whatever the bacteria we are having, they have destroyed through different factors. Then no micronutrient will be synthesized, no minerals will be synthesized, and no short chain fatty acid will be synthesized. So ultimately he or she will be suffering from the deficiency of vital micronutrients, and hence so many different kind of disorders and disease will arise in his or her body. So the colonization of each different section by different microorganisms can impact the local environment, and thereby positively or negatively influence the bioavailability of micronutrients. Suppose the duodenum, jejunum, ileum, colon, all these are designated to synthesize something. Like if they will all together synthesize only vitamin B12, then what will happen? Vitamin B12 will be synthesized in an excess amount. So anything which is below the optimized level or above the optimized level is always harmful. We require an optimized level synthesis. So duodenum, the microorganism who are residing in duodenum will have to synthesize what they are designated as. Like they only have to synthesize this one, and jejunum have to synthesize A, D, E, K, B vitamin, vitamin C, and also the quantity is decided. How quantity is decided? Because of course, suppose this is the duodenum, so it will have bacteria B1, B2, B3. So let's say B1 is of 70%, B2 is of 60%, B3 is of 80%, and likewise. But it is not necessary that all these three bacteria will be there in the jejunum. If they are there, the percentage will definitely vary. Like if B1 is present, it will not be like as in the duodenum 70%, it will be like 10%, 20%, 30%. So because there should be a balance, that duodenum will synthesize only this much of metabolite, jejunum will synthesize only this much of metabolite. So if any imbalance or dysbiosis will occur, then it may lead to any kind of metabolic disease. So for a normal person, I am talking about, if I am saying normal person, then you should be knowing that I am talking about a good gut health. I am talking about a good person having a very good health, good gut health. Good gut health means a very healthy microbiota is there, and all the microbial groups or the microbial diversity is in very proper limit. So they will synthesize whatever micronutrients which are required to perform the normal metabolic machinery of an individual. So this diagram shows that for every microorganism there is a limit that you have to synthesize this, you have to synthesize that. Suppose if we are having some function at our home, so whatever be the member, we designate them that you will take care of this part, you will take care of this part, that part, this part, that part, and likewise. So here also every part microbiota is designated that you will perform this, you will synthesize this, you will synthesize that, so that no, nothing will be in an imbalanced state. So next part, humans cannot synthesize all required micronutrient, they are unable to synthesize all the micronutrients, but they can synthesize if the microbiota is there. Microbiota will be there in the gut, so they will synthesize the metabolic products, and it will play role in different functions. So if, and for this reason also, if somebody is having bad gut, they are prescribed good probiotics. Exogenously they can be taken good probiotics to do this thing. If a person is, and he or she is not having a good microbiome, then dietitians may prescribe a good probiotic having a good composition of healthy gut bacteria. So he or she may take it to maintain the normal metabolic machinery, and then they will normally synthesize the metabolic product, so there will be no deficiency of vitamins and minerals. So we have to take, if our body is not producing micronutrient, so these are the sources from which we can derive the micronutrients. Like we can take dietary supplements, like many of us take oral supplements, and also we can enrich our gut with bacteria who are capable to synthesize some micronutrients. So this will be the best option, because this will be costly. Not only just costly, they may have some side effects, some sort of side effects will be there. So it is always better to do it by ourselves. If our body will have some bacteria that can synthesize different kind of micronutrient, so only we have to provide such environment, healthy environment, no acidic, no over alkalinity. So we can, if we will provide that, our bacteria will help us to synthesize the micronutrient, and no such deficiency will occur in future. So the common cell gut bacteria supply their host with essential nutrients and are the least explored resources for acquiring micronutrients. So whatever sources I have mentioned, 90% people are dependent on the exogenous sources, like they take the oral supplements. So because obviously their gut is not normal, after an age our gut microbiome disturbed, dysbiosis may occur. So we are dependent on the dietary intake, and also some oral supplement. The most common one is oral supplement. Many of us take oral supplement, like calcium deficiency is there, we take calcium tablets, vitamin B12 tablets. So likewise, but we are not exploring the most vital tool who can synthesize micronutrients on its own. Like we are not focusing on the gut bacteria, or you can say we are not focusing on the microbiota which is naturally present in our body. So nowadays, even dietitians also, and so many different people having the expertise in gastrointestinal disease are focusing on the gut health. They are focusing on to improve your gut health in order to be healthy. They are not just only prescribing the supplements, but they are saying that you should maintain your good health, good gut health, so that good bacteria will be there, the composition of the good bacteria will increase, and ultimately they will synthesize the vital nutrients, and it will be for a longer run, not for only 1 to 2 months. If you will maintain your gut health for a longer period of time, they will provide you all the vital nutrients which are required for the normal functioning of the body. So only we have to provide that condition. So gut microbiota is considered an effective bioreactor in the human intestinal tract, transforming various compounds into beneficial or harmful metabolite, thus having a crucial role in their bioavailability. Now, we are considering gut microbiota as an effective bioreactor. Now, I can tell you this point in an industrial perspective. Being a uh, being an industrial microbiologist, what we do, we exploit the natural property of a bacteria. Many, I'm talking about bacteria only, because out of bacteria and fungi, industries prefer mostly the bacteria. Why? The basic difference between bacteria and fungi is the basic, you can say the pros and cons. So the pros are bacteria only take overnight time to grow, but fungi require 48 to 72 hours minimum. But bacteria can be grown in 12 to 24 hours is sufficient. Bacteria is cool, they grow very fastly. So this reason, and their nutritional requirements are very simple, and they are easy to cultivate as compared to the fungi. In fungi there are so many things which we have to take care of, moisture level and many such nutrient composition we have to take care of. But for the bacteria, it is very good organism, we can grow it quickly in any media, in simple nutrient composition media. So industries prefer the easy thing, not sophisticated thing, because for an industrial perspective, they require an easy, cheaper, and hassle-free thing that did not require any sophisticated instrument, because they have to synthesize a product at an industrial scale in large amount. So obviously they will choose the simpler one. And also our nature favors the simpler thing, not sophisticated. Nowadays people are realizing that no such sophistication is required, go simple. So what I was talking about, we explore the function and role of bacteria in each and everything. Like you can take any bacteria, the only thing is that you have to take non-pathogenic, because obviously non-pathogenic is important, because if you will take any pathogenic strain, you will get infected by that one. So we take non-pathogenic strain, and a strain which is widely available. You don't have to strive for it. It should be Bacillus species. Bacillus is the most common and most widely used genus for an industrial perspective, because they have a brilliant capacity of extracellular protein production, of extracellular any such biomolecule they can synthesize. If they are getting the suitable media, suitable nutrients, and a suitable condition, they can synthesize anything, and they are non-toxic to us. Like you can say they are not non-toxic 100%, but majority of bacillus species are non-toxic. They may not cause any such disease to you. If you are having normal biosafety level cabinets also, BSL2 also, you can work on it. But if you are taking pseudomonas like I do in work in pseudomonas, so I require BSL-3. So you can't, a normal person cannot handle Pseudomonas. It is not recommended ethically also. So gut microbiota can be considered as a bioreactor. Bioreactor means they can be cultured in a fermenter to produce different kind of metabolites. Like I have said that the gut microbiota means what? They are having different kind of microorganisms, different group of microorganism, and they are capable of synthesizing micronutrient. Like they can synthesize short chain fatty acid, they can synthesize vitamins, they can synthesize minerals, and a lot more. So if we produce this on a larger scale, so for a larger scale, obviously we will need a fermenter, a bioreactor. So we can scale it, and then we can provide it to the people who are having such kind of deficiencies. So uh, industry industrial role is there. So nowadays, people are exploiting this property of a microorganism. They are culturing microorganism. They are providing some good amount of nutrient to them in a bioreactor. And nowadays, bioreactor is an automated machine. You don't have to stand there and observing each and every parameter, that which parameter is shooted, which parameter is not working properly. No, you only have to switch it on. Every parameter is automatic. It will be switched on, it will be switched off as per the requirement. You don't have to keep an eye on it 24/7. No, everything is now automated in an biotechnology world. So you only have to feed the media. Only one time you have to feed the media, and automatic sensors are there. If the level will decrease or go beyond the recommended limit, it will operate by itself only. You don't have to do any such human intervention is not required. So bioreactors are there, they culture in a very large amount. So whatever they produce, they just separate it, they purify it, they recover it, and uh, they make the final formulation, that whether it should go in a liquid form, or if liquid form is not required, they lyophilize it. It means they convert it into powder form. So what I am trying to explain you is that gut microbiota, not just gut microbiota, but this is an industrial perspective, that they are exploiting the role of microorganism in producing different kind of bioproduct. In bioproduct, you can say antibiotic, enzymes, biofuels nowadays, second generation biofuel. So all these things are possible because of the microorganism. So it is highly recommended to do such industrial bioproduct thing, internship and all those things, because this is need of the hour. With the advances of metagenomics technologies in microbiome research, there has been an increasing interest in understanding the contribution of different bacteria to human micronutrient status. So many in vivo studies and clinical trials addressing the association between bacterial classes or families with the host micronutrient balance. It means there are so many researchers, they are still trying to prove that there is a direct correlation between the gut microbiome and the host micronutrient balance. They have so many researchers have concluded with their studies, like in vivo studies and clinical trials, human subjects. So they have said that there is a direct relation between the gut microbiome and the micronutrient balance of the host. If the gut microbiome is good, then no such deficiencies are there. It means they are directly related, they are producing the metabolite, and therefore there is no such deficiency. So there are a lot more still needs to be unraveled to exactly told the mechanism, that how the microorganism is responsible for generating a particular kind of metabolite in the person. So it may have some therapeutic benefit. Already people are going in the human subjects, they are already on the clinical trial, that this particular metabolite is synthesized by this particular bacteria, and this, if we are consuming that metabolite, we will not having any kind of vitamin deficiency, micronutrient deficiency. So there are so many different pharma companies, they are trying their best to make such product available for normal people, because in India also we are having so much of micro nutrient deficiency cases, and people are avoiding it. Even I am too. So the gut microbiota produces significant quantities of a wide range of vitamins, notably vitamin K and B group vitamins. So if you are asked a question that a gut microbiota synthesizes which vitamin in majority, so it is the vitamin K and vitamin B group. Apart from these, all the other vitamins are in a minor quantity. So also the through the human microbiome project, which provided the high resolution of the human gut microbiota, which enabled a wealth and of in vitro studies evaluating the bacterial metabolic capabilities. So everything you can confer it through the in vitro. If you are not doing any in vivo experiment, at least in vitro experiments are required. You cannot say without doing in vitro just on a paper basis. No, at least you should go up till in vivo. But if in vivo is not possible, you can go into the in vitro studies. So there are so many studies. People are checking that whether the bacteria is capable of synthesizing such metabolite or not, if they are providing some nutrient. If this is a bacteria, and if it is cultured, if they are cultured in a nutrient, whether they are able to synthesize the vitamin K, vitamin B. So if they are able to synthesize, then they transfer this bacteria in an in vivo model, let's say mice. And then also, if it is producing the same thing, then also you need the third level of trial, that is in the human subjects. So you can say in vitro it is proven, in vivo in mice also, like mice is preclinical, and after that this is the clinical subject. So before going on to the clinical, you should have a clearance from the preclinical trial. The ethical committee have a level of doing the uh, all these things. So the if the mice are having the same as the in vitro, then you can go for the human subject. So if all the three levels are passed after a several trial, then the product is released in the market, and it is only just because of the human microbiome project. So you should know about the human microbiome project, because through this only we are able to know the composition. Composition means what kind of bacteria are there or microorganisms are there in the whole microbiome. So only because of the 16S R RNA sequencing, that is a part of human microbiome project, or metagenomics, we are able to know that in our microbiome A kind of bacteria are there, B kind of bacteria are there, C, D, and likewise. So all thanks to human microbiome project, that we are knowing what each and every group of bacteria of a human microbiota. So human gut common cells such as bacteroids, enterococcus, bifidobacterium can synthesize vitamin K and most water soluble B vitamins. So they have the capability that they can synthesize all these things by themselves. So their this property can be exploited on a larger scale, because people are having deficiency of micronutrient, vitamin deficiency, and all those things. So if we can make a product, we can definitely give them to meet their nutritional requirement, micronutrient requirement. Now, before answering this, you should know about the basic thing. If I am saying microbiome, I am talking about the collective genomes of the microorganism in a particular environment. But if I am talking about microbiota, if I am using while explaining microbiota, you should know about the community of microorganism themselves. If you have noticed, many a times I said microbiome microorganism. because microbiome is not microorganism. If we are saying microbiome, it means we are referring to the collective genomes of whatever microorganism are there in the microbiota. So for this reason I use microbiome microorganism to make it more clear. So you can say microbiota means the community of microorganism. If you have to say about microorganism, you will use the term microbiota. And if you are referring to the genome of the microorganism, then you will not use microbiota, you will use microbiome. So like genomics, so microorganism plus genomics, microbiome. Now come back to the question. Which of the following phyla is the most dominant in the human gut microbiota? Cyanobacteria,
B.
Bacteroidetes, Proteobacteria, Actinobacteria.
Bacteria do.
B, ma'am. Answer is B.
Answer me.
Bacteroidetes.
Hello, is anyone there?
Yes, ma'am. Answer is D. Am I audible to you?
Okay, so I'm unable to listen. Which of the following phyla is most dominant in the human gut microbiota? Cyanobacteria,
Bacter
Bacteroidetes,
no bacter
Proteobacteria, or
bacteroidetes.
Actinobacteria?
Bacteroidetes. Bacteroidetes.
It
Bacteroidetes.
is the Bacteroidetes, which is the most dominant in the human gut microbiota. Now next, I'm just curious to know that is anyone there or not. 18 people are there, but nobody is answering.
We're answering, ma'am. Is it not audible?
I answered, ma'am.
No, it is
We
not
all
audible. I
answered.
don't know why it is not
No.
audible. If I
But it's transcribing in the transcript.
is not working, so let me remove it. Hands free कर देते हैं. So am I audible?
Yes, ma'am.
Okay. So let's get back to this one. So I'm little bit disappointed with all of you guys because there are 18 people right now, but nobody
Ma'am,
is
we answered.
subscribing. No, not for
We answered.
that also. Yes, yes. That I got it, that my microphone is not working. 18 people are there, but nobody is subscribing bio.com. I'm feeling so disheartened that I am putting my so much of effort.
I subscribe, ma'am. I
Ma'am, subscribe
subscribe.
first day only. Yes,
Oh,
subscribe.
I subscribe.
okay.
I followed your videos.
Oh, thank you so much. Thank you. Okay, so get
Welcome,
back
welcome.
to this question. Germ-free animals are useful in studying gut microbiota because before answering, you should know about the whole concept that why we use germ-free animals to study the gut microbiota. Because now study the
Answer is
options.
C.
They lack any, yes, right answer. They lack any microbe and serve as a blank slate. So in microbiology or any such experiment, we always go for no organism template or no organism control, not or knock. Because if you are comparing anything with your microorganism, then there should be a control in which there will be no organism at all. So always in the in vivo study, if we are having that a mice is having some uh, microbiome, then there should be a control mice in which there is no such microorganism will be there, no microorganism will be there. Then only you can compare that if any organism will have a microorganism composition, whatever be it is. So a comparative study, then only you, with great confidence, you can say that if there is a group uh, of mice who are not having any kind of microorganism, they are germ free, and there is another group, treatment group, in which there are a lot of microorganisms, so there should be a always a normal control, no organism control, or in molecular biology we say no template control. With in PCR we run NTC. Now next one, the gut brain axis refers to what? The anatomical connection between spinal cord and intestine, neural signaling between liver and stomach, bidirectional communication between gut microbiota and the brain, hormonal control of gastrointestinal movement.
Answer is C.
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 You all are brilliant people. Yes. The right answer is bidirectional communication between gut microbiota and the brain. So this type of questions can be frequently asked from the topic gut microbiome and health. Don't worry, we will also have a session in which we will only solve some multiple choice questions, because as per my opinion, if we are not studying some subjective thing but we are going into the objective, then through objective you will be getting more about the conceptual thing, because what people are asking you should know about it very well. So always try to solve some questions from different examination paper, or from anywhere. There are so many sources. Now, gut microbiome micronutrient interaction, because this is an important part of digestive system, we have to focus it in a bit more detail. So you all know microbes utilizes micronutrient for their own growth and biological functioning. It is therefore not surprising that the intake of micronutrients can influence the compositional and functional structure of the gut microbiome. Now, one question arises here that I was having my RPC in the previous month. So my RPC member have asked me that I used a very this for this I am explaining. Black screen. So what I supposed to do is I work in NB, normal nutrient media, but I use Pseudomonas. So Pseudomonas always prefer to grow into a selective media which is rich in micronutrient, like so many iron, copper, manganese, and a lot more. So why I am not using this in my experiment? Because I am working with heavy metal remediation. So if I will use this media, this media is known as BH media, Bushnell-Haas medium it is. So I cannot use BH medium. So one of my RPC member asked me that why you are using NB if Pseudomonas is growing very like they require some micronutrient rich media, and along with that they require glucose, 2% glucose. It is optimized for the Pseudomonas that they require the Bushnell-Haas medium with 2% exogenous glucose supplementation. Without this, they cannot grow properly. It means it will grow in NB. Of course, it is a bacteria, it will grow in NB, but the incubation period will be much, much longer. So one of the expert asked me that why you are using it. So I answered him that, sir, if I will use this BH medium, which is already having some metal, so many metals, so that will hinder my own remediation experiment, because I am working towards the cadmium and chromium remediation. So how I can use already the metal supplemented media? So for that reason I am using NB. So he agreed with me and he said, okay, so what you do to this NB? So I said, instead of 12-hour incubation, I go for 24-hour incubation, because 12 hours are not sufficient for the required OD. Like the mid-log phase is achieved in 12 hours if I will grow Pseudomonas in BH medium. But if I am growing it in NB, it requires approximately 18 to 24 hours. It depends on the inoculum level, what inoculum I am inoculating in the media. So mid-log phase 0.6 to 0.8 is achieved in a double incubation time of whatever I am having in BH. So here comes the point that if you are saying that you will use microorganism to produce micronutrients, right? But microorganism microorganism also require micronutrient for their growth. So if they will synthesize, suppose they are synthesizing, if they are synthesizing 10 grams, just for an example purpose, 10 grams of vitamin K, and they require 1 gram for their own maintenance and all the dietary requirement of whatever a bacteria needs to grow. So if they are consuming whatever they have synthesized 10 gram, and out of those 10 gram they are consuming 1 gram, so total will be 9 gram. They have synthesized 10 gram, but they have utilized 1 gram. They have consumed 1 gram by their own community. So only 9 gram will be available. So this is known as bioavailability. Bioavailability means whatever a bacteria or a microorganism is synthesizing, but is it available completely to you for your use, or a part of it is available? So that is known as bioavailability. So this also plays a major role, that if the bacteria is consuming most part of whatever they have synthesized, then we require more such metabolites to be synthesized by the group of microorganisms. So a dietary supplementation with vitamin B, C, D, and E largely contributes to the microbiome composition by favoring the expansion and colonization of the intestinal mucosa by beneficial genera like Bifidobacterium, Lactobacillus, and Roseburia. So, if you are feeding your body with a particular kind of nutrient, then only a certain group of bacteria will grow. For example, if you are providing carbohydrate rich diet, then the bacteria who are capable of degrading carbohydrate will grow, not the protein rich bacteria will grow. So the population of, or the microbial diversity in your microbiota or your microbiome microorganism depends on what you are feeding to your body, what kind of substrate you are providing to your body. So accordingly, that group of bacteria population will be higher, like Bifidobacterium will be higher, Lactobacillus will be higher if the area or the organ is rich in vitamin B, C, D, and E. So if these 4 vitamins will be there in an area, then there should be a higher amount of Bifidobacterium, Lactobacillus, and the another one. And minerals such as calcium, iron, zinc, magnesium, and phosphorus can also shape the human gut microbiome. Obviously, these are micronutrient, and micronutrient play a very important role. If you are preparing any media and you are not putting any iron in it, if any selective media you are preparing, then no microorganism will grow in it, because uh, despite having so many different kind of carbohydrate source, protein source, bacteria will always respond to a micronutrient thing in your media, whether you are having the correct micronutrient or not, and the composition is also correct or not, whether it is increased or decreased will influence the growth of microorganism. So we always go for the optimization thing. In microbiology, nothing go quickly, like people think that this is very easy. No, we have to optimize each and every condition, because you never know how the bacteria will respond. If you are not having an optimized media, an optimized incubation time, an optimized pH, temperature, even the shaker RPM, that how much RPM does it require, whether it require the stationary kind of thing or the shaking, non-shaking. So all you have to optimize it. So likewise in our body also, our body is what? We are in vivo model. So microorganism requires that condition, like whether it require the carbohydrate rich diet or the protein rich diet. So that is why we are recommended to have a balanced diet. A balanced diet means a good amount of carbohydrate, good amount of protein, and also don't forget about the micronutrient, vitamins, minerals. We have to take green leafy vegetable, we have to take salad, and whole grain and mixed grain, so fruits. So all those things will have an adequate amount of nutrient and macronutrient as well as micronutrient, and that will support the good gut bacteria, and they will synthesize in return. What they will give in return as a return gift? They will synthesize the necessary metabolic product, so there will be no such deficiencies in your body. So high calcium intake has been associated with a high proportion of Clostridium cluster in men. Iron supplementation could induce a depletion of Bifidobacterium and an increase in lactobacillus level in children. So you can see, if we are taking some kind of supplement, then I said that it may or may not have some side effect. So this is the side effect. If we are taking, suppose you, if your body is having vitamin B deficiency and you are taking medicine to remove such deficiencies, then it is definitely, it can be there is a probability that the other bacteria population might be affected. That here it is mentioned, if iron supplementation would induce a depletion of bifidobacterium and an increase in the lactobacillus. Lactobacillus will increase and bifidobacterium will be decreased. So all those things have a impact on the normal microbiota, normal microorganisms also. Like if you are treating any one disease, because we know this is the whole gut, and you are having microbiota, everything is colonizes by the different kind of microorganism, like bifidobacterium, clostridium, lactobacillus. So if you are taking supplement, that supplement will be absorbed in the whole epithelium. So if one population is influenced, one population is increased, there is and chances that the other population will decrease. So there is a probability of both the things, like bifidobacterium will be depleted, lactobacillus will be increased. So and also you can see phosphorus supplementation. Phosphorus supplementation can lead to an increase in microbial diversity and level of short chain fatty acid in stool. So you have to take care of, doctor always prescribe such medicines which will not cause a greater impact on the other group of microbial diversity. Because if you are taking a medicine for a certain thing and it will cause harm to some other good bacteria, then what's the point of consuming that medicine? So they always take care of the other microbial diversity which is present in our gut. Now, on the other hand, it has been shown that various members that make up the intestinal microbial ecosystem can influence the bioavailability of this point I was talking about. If the microorganism will itself consume some amount of metabolite or micronutrient, then the bioavailability will be definitely influenced. 10 gram वो खुद बना रहा है. If the bacteria will consume 1 gram, then only 9 gram will be available to you for your use. So we have to control their absorption. So for this reason, so many body adopt so many different kind of methods we will study. So particularly phosphorus and calcium and synthesizing vitamins such as vitamin K and water soluble fraction of vitamin B. Now see what our body do. how to explain this point. Suppose if we are having so much of cash at home, so always a robbery or a theft or such kind of thing is always in our mind, that oh no, we have so much of cash, anybody can steal it. So what we do, we just go to the bank and we deposit it. This is a safe option. So likewise, whenever you know that your bacteria has synthesized 10 grams of metabolite xyz whatever it is, so readily our body do what, there are so many different kind of, I said, enzymes. They will modify the enzymes by putting so many functional group over there. So now bacteria will no longer be able to use that, because suppose bacteria can use only suppose this is the triangle, this is the metabolite. So bacteria can very well consume this metabolite. But immediately after the synthesis of this metabolite, our body will do what? Our body will modify the outer surface of this. They may add some functional group or some any other thing to it on its external surface, so that it will not be recognized by the microorganism, and microorganism will not recognize it, and the microorganism will not consume it. So he will not consume the 1 gram, and total 10 gram will be available to you. I hope this point is clear. Like, our body is so smart. Our all the immune cells and all the body cells work in a very coordinated manner. They are very, very smart. You can't even think of. So they will think that if 10 gram is synthesized, they immediately modify that with different enzymes available in our body, so that bacteria will not consume it. So the bioavailability will be increased. Like, I have also worked in one of my research experience is also in nanobiotechnology. So the yield is very low. In very microgram we get the yield of the nanoparticle. So we always go into the method which will increase the bioavailability of the nanomaterial. So our body also adopts such measures to protect and to preserve whatever our microorganisms are synthesizing. So all these things are uh, very well executed if your body homeostasis is maintained. If somehow you have destroyed it, nothing will be in a balanced state, dysbiosis will occur. So it's already 9:58. Okay, we will see this diagram and we will conclude. So this is the diagram in which you can see the gut microbiome micronutrient interaction. Like, there are minerals, there are short chain fatty acid, and representative are butyrate, acetate, propionate, vitamins. So everything is synthesized by our gut microbiome. And you can see these are through dietary intake and these are through supplements. There are so many exogenous supplements also nowadays in the market, and the natural way is to consume fruits and vegetables, or else you can use the exogenous supplementation. So what they do, they enrich our gut with good bacteria, and they synthesize the minerals, short chain fatty acid, and vitamins. Three major products they synthesize out of their metabolic machinery. So we will see in depth tomorrow. So this is all for today. Let me first stop the class recording.